Welcome to WIP's Biochemistry, and this time we will be discussing the metabolism of one of the most important amino acids, namely glycine. Now, glycine, as we know, it's the simplest amino acids and it is optically inactive because there is no presence of chiral center or asymmetric carbon atom is not present in glycine, obviously. So that's why it is optically inactive. Now, glycine metabolism glycine can be considered as a metabolic junction. Now, just like oxaloacetate or pyruvate or alpha-ketoglutarate, which give rise to a number of molecules, and they are formed from a number of molecules also. So is glycine also. It is actually the precursor of some of the important molecules like purine or heme and so on. Now, glycine is the most abundant amino acid normally excreted into urine, and nutritionally it's a non-essential amino acid, and metabolically it's a glucogenic amino acid also. And glycine is very important because it is one among the commonest amino acids found in protein structure. Exemplified by collagen. Now, collagen contain very high, almost 30% of glycine, which means every third amino acid in collagen is glycine. Now, glycine can be formed from a number of metabolic molecules, biomolecules, especially from some amino acids also. Now, basically, we are dealing four different types of synthetic pathways for glycine here. After that, we will go to see the degradative pathways of glycine also. Now, these are the four molecules from which glycine is basically formed. That is carbon dioxide and ammonia. Then from serine, an amino acid, threonine, another amino acid, and from glyoxylate. So we will see this one by one. So this is the obvious biosynthetic pathways of glycine. First of all, it is formed from carbon dioxide and ammonia, and the additional carbon in glycine is contributed from N5, N10 methylene THF, which is a one carbon unit. It is present in the cytoplasm as cytoplasmic pool of one carbon units. Glycine cleavage system is involved. We will see in detail later. Now, here there is an amino acid, another one here, serine. Actually, it gives rise to glycine with loss of one carbon. So the extra carbon in serine is received by tetrahydrofolate to give rise to N5, N10 methylene THF, which is the one carbon unit. The enzyme involved is serine hydroxymethyl transferase. Here it is threonine aldolase, which give rise to glycine, and acetaldehyde leaves threonine. So the extra carbon is lost as acetaldehyde. And this one, it is a transamination reaction, the interconversion of glycine and glyoxylate. Glyoxylate, obviously, it's a alpha keto acid. Here again, there is another alpha keto acid involved, and the enzyme involved is glyoxylate amino transferase. Let us see one by one. First of all, before that, you can go through this. This is just whatever I explained just now is written here. The first one is formation of glycine from carbon dioxide and ammonia. Now, glycine obviously is having two carbon units in it. One carbon is received from carbon dioxide. The amino group is received from ammonia, and the extra carbon is actually contributed by N5, N10 methylene tetrahydrofolate, which is the donor of the single carbon unit or single carbon in glycine. And the enzyme known as glycine synthase. It needs a number of coenzymes: NADH, lipoamide, tetrahydrofolic acid, and PLP, pyridoxal phosphate, also is a coenzyme for this particular enzyme, glycine synthase. It is actually this process is reversible, and the enzyme complex, the multi-enzyme system, it is not considered as a enzyme complex, multi-enzyme complex in the real sense of it. This is mostly described as a enzyme system or multi-enzyme system. Now, this system actually is often described as a glycine cleavage system. Glycine cleavage system also, which means this is actually reversible. Now, this is a formation of glycine from serine. Obviously, it's a hydroxyl group containing amino acid serine. the alpha carbon of serine is taken up by glycine, whereas the second carbon it is donated to tetrahydrofolic acid so as to form the one carbon pool of N5N10 methylene THF. As I said, this is actually one carbon unit. The extra carbon of serine is contributed to tetrahydrofolic acid to form N5N10 methylene THF. Now NADH is involved. The enzyme is serine hydroxymethyl transferase with pyridoxal phosphate acting as the coenzyme. Obviously, this is a reversible reaction. We can guess the glycine will receive the one carbon to become serine. That we will discuss later. Now, thus another amino acid threonine, which gives rise to glycine, and the extra carbon. This unit is it leaves threonine as acetaldehyde. You can just study here. This structure, it becomes acetaldehyde, whereas the rest of the molecule is forming glycine. So this is actually a non-reversible reaction, threonine aldolase catalyzed reaction, because threonine is an amino acid which is essential. It is an essential amino acid, cannot be formed or synthesized in human beings. So this is obviously it is irreversible. And this one is this is actually a transamination reaction. Obviously, glyoxylate can be formed from glycine, and glycine can be formed from glyoxylate. The amino acid, the amino transferase involved is glyoxylate amino transferase, which obviously all amino transferases act. This pyridoxal phosphate is involved in this process as coenzyme, and obviously glyoxylate can be generated from glycine also. Now, so what we have discussed so far is synthetic reactions of glycine, which includes this one. This is carbon dioxide and ammonia, and serine. Serine can give rise to glycine, and glyoxylate can also give rise to glycine. And these three reactions are reversible. Now, obviously, glycine can be broken to give rise to carbon dioxide and ammonia with the help of glycine cleavage system, which is almost a reversal of glycine synthase system. And glycine can accept this methylene THF, one carbon unit from methylene THF to give rise to serine, as we saw. As also glyoxylate. Obviously, this glyoxylate glycine reaction is a transamination reaction. Obviously, it can be reversed to give rise to glyoxylate. So glycine from glyoxylate. So let us see these three reactions first. And obviously threonine, this is not a reversible reaction. Obviously threonine cannot be synthesized in our body, being an essential amino acid. So you can go through this. Then yeah. So this is the glycine cleavage system. Yeah. Glycine cleavage system, which utilizes pyridoxal phosphate. Now, obviously, all these coenzymes are involved in this process also, including lipoamide, NAD, and THF, methylene THF are involved in this reaction. And the enzymes, the glycine cleavage system actually includes the enzymes like glycine decarboxylase, which actually uses the pyridoxal phosphate, amino methyl transferase, which uses lipoamide, and lipoamide dehydrogenase, which uses NAD plus as coenzyme. So this is glycine cleavage system, and obviously the extra carbon in glycine is contributed to tetrahydrofolic acid to give rise to the one carbon unit N5N10 methylene tetrahydrofolate. Now, this is another of the reversible reactions we saw earlier. Glycine. Now, just as serine can give rise to glycine can be synthesized from serine, and serine can be synthesized from glycine also. And obviously, it is the one carbon unit which is the key molecule here. The enzyme is the same, being a reversible reaction. And here The synthesis of serine from glycine is important in another sense also, because serine can be converted to glucose. Serine can be converted to pyruvate, and pyruvate obviously is a substrate for gluconeogenesis. So this is a mechanism for production of glucose also through gluconeogenesis. And this one, obviously, as I said earlier, glycine and glyoxylate interconversion is a transamination reaction involving aminotransferase, glyoxylate aminotransferase, which utilizes pyridoxal phosphate as the coenzyme. So obviously glyoxylate and alpha ketoglutarate are the alpha keto acids involved, glutamic acid and glycine are the amino acids involved, just as in the case of a classical transamination reaction. Now, coming back to the overview of glycine metabolism, both the synthetic and degradative pathways are shown here and represented here. Now, glycine can be a precursor to a number of important biomolecules, which are shown here, glycine can give rise to purine, it can give rise to heme, as shown, and glutathione, and glycine can conjugate with bile acids to give rise to bile salts. Glycine can be broken down to creatine, and which will give rise to creatine phosphate, a high energy compound, and then creatinine also. So all these reactions we will discuss one by one. Now this is apart from the most important reaction of in which glycine is involved, because glycine is the commonest amino acid found in protein structure. So protein synthesis is the most important reaction of glycine, and glycine is mostly present in the interior structure of protein also. That may be mentioned here. Now let us see one by one. Now first of all, glutathione. Now glutathione, synthesis of glutathione. It's a tripeptide formed from three amino acids, namely glutamic acid, cysteine and glycine. Now the reaction goes like this. Glutamic acid combines with cysteine in the presence of glutathione cysteine ligase to give rise to gamma glutamyl cysteine, which accepts one molecule of glycine to give rise to glutathione. So the enzyme involved is glutathione synthase. So glutathione is also called gamma glutamyl cysteine glycine, representing the names of the three amino acids involved in its synthesis. Now gamma glutamic acid, because the carboxylic acid, glutamic acid is a dicarboxylic acid, monoamino amino acid. The carboxylic acid involved in this reaction is the gamma carboxylic acid, which is attached to the gamma carbon. It's not the alpha, the primary carboxylic acid. It's not involved in this reaction. The primary carboxylic acid is actually attached to the alpha carbon. But here it's the second carboxylic acid group in glutamic acid, which is attached to the gamma carbon, is involved in this reaction. So that is why this is called gamma glutamyl cysteine glycine. And glutathione is a reduced form. This is an important molecule in scavenging of free radicals. It can be oxidized to give GSSH also. Now these are the further reactions of glycine which we will discuss. It can be degradative for glycine, but at the same time synthetic for the molecules here, like heme biosynthesis, purine biosynthesis. So just go through all these and let us see one by one. Now first of all, the synthesis of heme. The first key reaction involved in heme synthesis involves glycine, the ALA synthase catalyzed reaction, in which glycine and succinyl coenzyme give rise to ALA, or aminolevulinic acid. And this succinyl coenzyme, we are familiar with this particular molecule. This is a substrate in TCA cycle, and this is an important reaction because this is the reaction which is catalyzed through, regulated through feedback. If heme accumulates, it regulates ALA synthesis through feedback. So this obviously involves glycine as one of the precursors of heme. Now, the whole molecule of glycine form the part of purine, as shown here. So this is 2 carbon and 1 nitrogen, which belong to glycine actually. So this whole molecule of glycine forms part of The basic structure of purine, and purine, as we know, adenine and guanine are formed from this structure. And the glycine actually contributes the fourth and fifth carbon, and this at the seventh position, nitrogen also is contributed to from glycine. So purine formation from glycine. Now, benzoic acid, which is actually used as a preservative, a food preservative, which is actually should be detoxified in our body. And this detoxification of benzoic acid, one of the pathway is using glycine. Now, glycine can be combined with or conjugated with benzoic acid so as to give what is called benzoyl glycine. This is also called hip uric acid, the common term, because it was first isolated from horse urine. So this is one of the important pathways of detoxification in our body of benzoic acid. And glycine can conjugate with bile acids. Now, bile acids are obviously formed by the degradation of cholesterol, extra cholesterol, which are present in our body, and this is one of the mechanisms to get rid of the extra cholesterol in our body. So cholesterol can give rise to bile acids, and bile acids can be combined with, for example, cholic acid can be combined with the glycine to give rise to glycocholic acid. So as glycodeoxycholic acid also. So these molecules can actually combine with sodium or potassium like this to give rise to sodium glycocholate or potassium glycocholate, which are called bile salts. And these bile salts are passed through bile juice into the duodenum, where it acts as an emulsifier for the emulsification of fats which are coming to duodenum or intestine. So there is a purpose for this formation of glycolates. So it is involved in digestion of fats. So that is the role of glycine in actually getting rid of cholesterol also here. Now, glycine can be a precursor for the synthesis of creatine also. Now, creatine can give rise to creatine phosphate. And the creatine phosphate is a high energy compound. It will be stored in muscle cells, and it can release energy on breakage at times of necessity. Creatine is actually synthesized from three amino acids, namely glycine, arginine, and methionine. It's actually the activated form of methionine, namely S-adenosyl methionine, which is involved in creatine synthesis. The first reaction occurs in mitochondria of kidney and pancreas, and it involves the transfer of guanidino group of arginine to glycine to give rise to guanidino acetate GAA. The enzyme involved is arginine glycine amidinotransferase. And the second step or second reaction involves the S-adenosyl methionine, the active methionine. It donates methyl group to guanidino acetate and form creatine. The enzyme involved is shown here. And this reaction occurs in liver. Now, creatine can give rise to creatine phosphate, the high energy compound, which it can give rise to creatinine spontaneously. And creatine and creatinine are different. Creatinine is a waste product. It's a product which has to be removed. It's a toxic product which has to be removed from our body, and the removal of creatinine take place through urine. The elevated level of creatinine is important in kidney dysfunctions. Now, so this is finally glycine is incorporated into tissue proteins. This is one of the important functions of glycine. It is widely distributed in protein, especially where there are bends and turns are there. And glycine is mostly seen in the interior of proteins also. And obviously in collagen, it is in plenty. Every third amino acid of collagen is glycine.